Okay. Oh, I wish I took that shot. That is a nice shot.
No,
the light comes on and then it goes off.
Oh, okay, it's just blowing, okay, it's just blowing balls, that happens all the time. All right, that's a light show, free light show. All right. Yeah, I see that.
Because it's right next to the picture.
Yeah. Hey, look, I did it.
Big, big, big.
Oh, crap, all right, doesn't have, I only have limited powers here. Okay. That's why from administration I can do that, you know. All right. Okay, let's take a look more at translational regulation. And the masters of this whole process are oocytes. And we can learn a lot about translational regulation in oocytes. Because think what an oocyte does. Recall when we were doing in the lab, recall we said oocytes are very large, complex cells. They got to have a food supply like yolk for the embryo, or endosperm if you happen to be a plant oocyte. And you have also a developmental program that's pre-stored in the oocyte. Once you activate that program, development will proceed to a certain point. As we'll see later, you can actually block all transcription in an oocyte, fertilize it, and it will develop in some cases all the way to the beginning of gastrulation before it stops. That's true, by the way, in sea urchin, so we may actually do a little experiment on that somewhere down the way in the lab, depending. Okay, but anyway, you can block transcription completely in oocytes, and it will still continue on to a certain point. Now, it turns out with mammalian embryos, that only about 2 or 3 cell divisions before it rests. But some embryos can go a remarkably long time, and that means that the program for development must already be stored in the oocyte. And what that means is the oocytes are going to store enormous amounts of necessary proteins plus necessary messenger RNAs. And these molecules are put in place during oogenesis, long before fertilization. As we are building up this egg, or this oocyte more properly, as we assemble this oocyte, we're putting together the molecules that will program early development. Now, when you look at messenger RNAs, you might ask your question, if oocytes are packed full of messenger RNAs that are going to do various developmental events, what keeps them from being translated before the right moment, meaning fertilization or appropriate time after fertilization? So, you can do experiments on oocytes to get insights into how we prevent translation until the right moment. So oocytes are loaded up with untranslated messenger RNAs. What do we see in many cases? We do see a lot of examples of incomplete 5' caps. That's pretty common. Not universal, but it's pretty common. So these messenger RNAs go in with an incomplete cap. Ribosomes will simply not recognize it. At some point after fertilization, we activate the methyltransferase that sticks the methyl group on the guanosine. And now they can be translated. So that's one type of thing. Blocking translation is another thing. Now here's a little experiment here. We take an oocyte. We suck out we suck out the messenger RNAs. We extract messenger RNAs from an oocyte. Now our control experiment, we inject those messenger RNAs into another oocyte and see what happens. Okay, and what happens is oocytes have very low levels of translation. Not quite 0, but it's really, really low. So your control situation is normal. Low translation. Now let's do another experiment. We take those messenger RNAs this time, And what we're going to do is expose them to high salt, like 0.5 molar, even 1 molar salt. Now, why do you put them in a whole bunch of briny water? Because high levels of salt tend to strip proteins off of things. For instance, my current grad student, Ms. Linda Major, she's isolating the histones from plant cell nuclei. The way you do it is you take these nuclei, mash them up, and then expose them to very high salt. That strips the proteins away from the DNA. You can get rid of the nucleic acid, and then precipitate out the proteins. And hopefully she's going to have a whole bunch of them. I'll find out after class. Okay, so high salt strips proteins off of this. So now we expose these guys to high salt. And then collect the messenger RNAs again, get rid of the proteins. And now put them back into an oocyte. When we do this experiment, lo and behold, those messenger RNAs put back in the oocyte with the protein stripped off of them, the translation rate goes up significantly. That's telling us something. That's telling us that ordinarily in oocytes, many messenger RNAs are bound to proteins that inhibit translation. If you remove those proteins, translation goes up. All right, that's another kind of thing we can do. Question?
No.
Oh, okay. A third thing we can do is regulate translation factor. Now here's some experimental evidence that is another thing that oocytes tend to do. You can do something called cell-free translation experiments. And you can buy these things in a kit form. What it is translation in a test tube. For a couple $100, they give you a vial full of ribosomes, tRNAs with amino acids, GTP, there's an energy source, a whole bunch of goodies. Now when people first came up with the idea, could we get translation to occur in a test tube? What they did was they would isolate ribosomes, they'd isolate tRNAs, they'd throw in an energy source, they'd throw a messenger RNA, they'd throw in nucleotide triphosphates, the building blocks, and they put all that stuff in the test tube, mix everything together, and look, and lo and behold, absolutely nothing happened. That was the first attempt, and then somebody came up with the idea, maybe there's some kind of unknown things in the cytoplasm that are required for translation. So they threw in some cytoplasmic extract, and you got translation in test tube. That's what we call cell-free translation. You buy these kits, and what you have here is some kind of cytoplasmic extract containing translation factors. So we have this kit, all you have to do, amino acids, yeah. You have this little kit, and like I said, you could buy these through biological supply houses for a couple $100, these cell-free translation kits. All you got to do is add messenger RNAs of your choice, and lo and behold, translation will occur in a test tube. Okay, now, people did a little experiment here. They threw everything but the cytoplasmic extract, and now what we're going to do is make our own cytoplasmic extract and throw in some kind of messenger RNAs and see what happens to translation. Okay, so what we're going to do first is we're going to take oocyte cytoplasm. We put in our cell-free translation system, and we add our messenger RNAs, and we find very low translation. Not much happens. It's hardly any better than having the kit without any cytoplasmic extract. So it's saying that oocytes have either very low levels of translation factors, or those translation factors are regulated in such a way that they are not active. In either way, oocyte cytoplasm does not support translation very well. And you have a question, Ms. Killam?
Yes. Okay, so it goes from, to the cytoplasm, is that arrow pointing to the GPP or?
No, it's pointing just this whole cell-free translation system.
Oh, okay.
Okay,
that
so in
whole
other words-
system, and then that whole system equals.
Yeah. So in other words, instead of using the store-bought cytoplasmic extract, we'll add our own cytoplasmic extract instead. And the take-home lesson here is if you take extract from oocytes, that doesn't work very well. That tells you that either you have very few translation factors in there to begin with, or what ones you have are inactive, or both. But oocyte cytoplasm is terrible for supporting translation. Now let's fertilize the egg and take egg extract. And we take once again our cytoplasm, throw it into our cell-free system here, and what happens? Whoa, we have translation up the wazoo, folks. We have a lot of translation. That's telling us that once that oocyte is fertilized, something is happening about translation factors. Either we have a whole bunch of them being produced, or more likely, they were always in the cytoplasm to begin with. Don't forget, there's not much transcription right after fertilization in early fertilized embryos. Transcription levels are very low. So most likely what we have here is we have active translation factors. Here we have regulated inactive translation factors. It's more likely that we're dealing with regulating these things rather than all of a sudden at a moment's notice cranking out and producing huge amounts of new translation factors. They were probably already there, but they were just in an inactive state. So that's another kind of backup thing we see. We've got a situation where you have sequestered messenger RNAs. They're tied up to the oocyte cytoskeleton in places ribosomes can't get to. We coat these messenger RNAs with translation inhibiting proteins. Strip them off, translation goes way, way up. We have a whole pile of inactive translation factors that we activate only after fertilization occurs. So we have a whole bunch of different things that inhibit translation in oocytes. Plus we have a lot of messenger RNAs that have partial incomplete 5' caps. That means ribosomes, no matter where you find them, are not going to even know they're there. Okay, so another thing we see too is if you take total messenger RNA from oocytes and you measure how long the poly A tails are, and you take total messenger RNA from eggs after fertilization and measure their poly A tails, you know what happens? The poly A tails get bigger. Yeah. The poly A tail, the average poly A tail length grows significantly larger right after fertilization. And what happens is, it turns out, if you measure the activity of our cytoplasmic poly A polymerases, these are what build up poly A tails, you find these guys are active as all get out after fertilization, yet they're not very active before fertilization. Now, a little sidelight to this. In many cases, in oocytes, it's not necessarily true of regular cells, but oocytes, don't forget, they're specialized for storing huge amounts of untranslated messenger RNAs and translating them only at the appropriate point after fertilization. Well, what happens a lot of times is the mRNAs go out into the cytoplasm, And then we cut and remove a good chunk of the poly A tail. Practically right after they exit the nucleus and go in the cytoplasm. In oocytes, we have enzymes that chew up the lion's share of the poly A tail in these guys. So these guys start out in oocytes with very short poly A tails. Recall, we said you remove the poly A tail entirely, translation drops 90%. So already we've really reduced translation efficiency. Now somehow we're going to protect these guys from nucleases regardless. There's other factors that can do that. But they come in, they're stored with short poly A tails that are very inefficient promoting translation. And then right after fertilization, we fire up the polymerases, lengthen those poly A tails to what they're supposed to be, translation starts skyrocketing. So we see that kind of thing. Oocytes have extremely low levels of translation, but right after fertilization, translation just goes through the roof. It skyrockets. And these are some of the reasons why. We have several different mechanisms, each one of them playing important roles in regulating translation. Okay, that kind of overviews some of the aspects of translational regulation.
Before you erase more, this slide, picture that you're here, this mRNAs, they go out, and then you remove most of the poly A tails, so then they start out with a shorter tail.
Yeah.
Well, then where is it pointed to now? Like, where is like the,
After fertilization.
Oh, after fertilization, yeah, after fertilization, then we lengthen the poly A tails.
okay.
Okay. All right. Okay, so those are some examples of translational regulation.